So should I start?
Yes.
Good morning, everyone. Today I'm presenting a seminar on the topic epigenetics. So epigenetics is the field of study surrounding stable alterations to the DNA and histone proteins that alter gene expression. Epigenetic modifications are responsible for tightly regulated tissue and cell type specific gene expression patterns, and aberrations in these expression patterns can give rise to certain diseases, most notably some types of cancer. Environmental factors can influence the expression of genes within a cell through modifying epigenetic marks without mutations to the genome. These environmental changes to the epigenome are so strong that even monozygotic twins can be identified by examining their unique epigenetic patterns. Changes in epigenetic patterns can cause an alteration of gene expression, which in itself can have many downstream effects, including changes in disease risk, metabolism, and stress response. Epigenetics was coined by Conrad Waddington in 1942, and it was fused by forming the terms genetics and epigenesis. Epigenesis is an old term used to describe the developmental program by which the zygote gives rise to different cell types in a multicellular organism. This represented the cooperation between genetic traits and environmental conditions that together established a stable cellular phenotype. The notion that non-genetic factors play an important role in development was further formalized by defining epigenetics as a stably heritable phenotype resulting from changes in a chromosome without alterations in the DNA. The epigenetic code consists of histone proteins, histone modifying enzymes, structural proteins, DNA modifying enzymes, ATP-dependent chromatin, remodeling machinery. It is a 3D structure, and the cellular content is unique for each cell type. It may change in response to environmental cues and cell cycle progression. Rapid changes in response to environmental changes are seen, and they could also be long-lasting. They can also modify gene expression and are sometimes heritable. Now let's talk about the mechanisms. Firstly, the histone modifications. The modifications of histone proteins is brought about by histone acetylation, histone methylation, which could be activating or repressing, histone phosphorylation, and DNA methylation. So these are the histone proteins. And so this is the process that it condenses the chromatin and causes transcriptional repression. Then in phosphorylation, it opens the chromatin, causes transcriptional activation. In methylation, the chromatin is condensed or transcriptional repression occurs. So these are the mechanisms by which histone proteins are modified. Now let's talk about the environmental influences and epigenetics. Individual genetic background and environmental factors are intertwined to lifestyle in determining the health status of individuals. Increasing evidence shows that environmental and lifestyle factors may influence epigenetic mechanisms. Firstly, let's talk about nutritional epigenetics. Certain dietary bioactive food components can change gene expression via alterations in DNA methylation and histone modifications, a field of study that may one day lead to the development of epigenetic diet. Firstly, folate and vitamin B12 intake. Folate and vitamin B12 play an important role in DNA metabolism and are required for the synthesis of methionine and S-adenosyl methionine. Abnormal intakes of these during the prenatal and early postnatal life form the basis of developmental origins of health and disease, the Dohad hypothesis. Abnormal nutrition during prenatal and early postnatal life can also affect the behavior during adult life by epigenetic mechanisms. The downregulation of gene encoding relin. This is a protein involved in brain development, neurotransmission, memory formation, and synaptic plasticity, is found in the female offspring of mothers with DMD. DMD is deficient in methyl donors. That is, DNA methylation is not occurring properly. There is increased risk of schizophrenia in such individuals, especially with deficiency of folic acid. Previous surveys have found that up to 1/3 of psychiatric inpatients had folic acid deficiency, and this was associated with the presence of depression and dementia. Now, selenium. Studies have found that both high and low levels of selenium are linked with dysregulation of oxidative and inflammatory pathways, offering another mechanism that could explain the observed association between selenium levels and depression. Selenoproteins such as glutathione peroxidases, redoxin reductases, and selenoprotein P are known to provide protection against lipoperoxidation and oxidative cell damage. Now, low selenium concentration is associated with an increased level of pro-inflammatory cytokines, such as interleukin-6, C-reactive protein, and growth differentiation factor 5. A study in 2005 demonstrated an association between low levels of cholesterol and increased risk of depression and suicidality, suggesting a possible role for selenium's anti-lipoperoxidative actions in its protective effect against depression. Now, zinc. Evidence has demonstrated that several key enzymes and zinc atoms in the reactive center and binding site play an important role in DNA methylation and histone modification. Therefore, a zinc deficiency may disrupt these functions and result in epigenetic dysregulation. The balance of intracellular and extracellular zinc levels is crucial for maintaining zinc homeostasis in many brain regions, including those involved in pathophysiology of depression, such as hippocampus, amygdala, and cerebral cortex. Intervention studies in both humans and rodents involving dietary or supplemental zinc have reported antidepressant-like and mood-enhancing activities of zinc. Let's talk about environmental pollutants. In environmental studies, the flexibility of epigenetic states has generated growing interest in evaluating whether this environmental exposure can modify the epigenetic states, including, again, DNA methylation and histone modifications. Firstly, arsenic. In a study from India, significant DNA hypermethylation of P53 and P16 promoter regions was observed in blood DNA of subjects exposed to toxic arsenic levels compared to the controls. Then prenatal arsenic exposure has also been associated with changes in expression of soluble proteins like tyrosine kinase 1 gene, playing a key role in them, inhibition of placental angiogenesis, and related growth retardation in cord blood cells. Now, mercury. In a study, association between DNA methylation in newborn cord blood and prenatal mercury concentrations has been revealed, suggesting that exposure to mercury may lead to epigenetic modifications involved in altered immune profiles. Prenatal mercury exposure has been associated with placental DNA methylation changes and adverse infant neurobehavioral outcomes. Manganese, a micronutrient. Fetal manganese exposure resulted in differential methylation of several loci in the placenta, including those residing in fetal growth, neurodevelopmental, and cancer-related genes. Epigenetic influence of stress. Psychosocial stress involves neurophysiological changes resulting from the anticipation or perception of challenges to well-being that are located within the social environment. Recent research that epigenetic mechanisms are an important interface through which the body interprets and responds to stressful experiences. Antenatal and perinatal stress. This period is a time of rapid growth and development of the fetus and is also a period of heightened plasticity. Adverse experiences during this period have been known to induce significant effects on neurobiology, metabolism, and physiology that can persist across the lifespan. Antenatal stress or anxiety has a programming effect on the fetus, which can persist up to adulthood and result in an elevated risk of autism, schizophrenia, anxiety, depression-related behaviors, and also impaired cognitive performance in later life. Postnatal period is also a period of epigenetic plasticity. The epigenetic link between adversity in early childhood caused by exposure to emotional, psychological, sexual, and physical abuse, maltreatment, poor quality parenting, or loss of parents, etc., and later life health outcomes has been demonstrated repeatedly. Research has confirmed and extended the findings of animal experiments on the effects of this early life stress. Moreover, childhood abuse, neglect, and deprivation were also found to be associated with an increased NR3C1 promoter methylation. Exposure to early life stress results in enhanced susceptibility to disorders such as MDD, generalized anxiety disorder, schizophrenia, and autism spectrum disorder. Maternal lifestyle. Now, firstly, let's talk about maternal care. Maltreatment of mothers of nursing newborn offspring during the first week of postnatal life has shown to elicit repeated vocalized distress calls in offspring and long-lasting changes of methylcystosine and its derivative hydroxymethylcystosine at the BDNF locus in the hippocampus, amygdala, medial prefrontal cortex of exposed offspring during adolescence and adulthood. The paternal influence. A father's exposure to environmental factors can also play a role in an offspring's epigenetic patterns and health. Evidence suggests that sperm epigenetic modifications can survive reprogramming following fertilization and be inherited by the embryo. For example, it takes place in Angelman syndrome with paternal origin. Now let's talk about the chronic stress factors. Firstly, the low socioeconomic status. Studies on the impacts of low socioeconomic status on epigenome have reported altered patterns of DNA methylation in samples extracted from whole blood, fractioned white blood cells, or buccal swabs. Studies have shown that a low socioeconomic status is associated with variation in genomic methylation patterns, and mental health problems such as depression, anxiety, addiction are worse in more unequal societies, and these disorders are more prevalent here. A study has identified predictive links between these low socioeconomic status, wherein differential methylation of SLC6A4 serotonin transporter gene promoter elevated amygdala functions and symptoms of depression. These results thus suggest a plausible biological pathway through which low socioeconomic status by methylation of this gene could attenuate expression of the serotonin transporter, leading to increase in both amygdala reactivity and risk of depression. Maternal exposure to nicotine during pregnancy has causative relation to miscarriage, stillbirth, preterm delivery, apart from the metabolic, cardiovascular, and neurobehavioral disorders in offspring. Now, maternal smoking is also implicated as an associated risk factor in the etiology of ADHD in children. Nicotine exposure in prenatal period is also associated with development of Tourette syndrome in offspring, with comorbidities like tic disorder and obsessive compulsive disorder. Now, fetal alcohol spectrum disorder is a series of neurodevelopmental disorders affecting the attention, memory, craniofacial development, auditory, motor function, and language problems in children associated to maternal gestational alcohol exposure. Malnutrition. Supplementation of folate, as previously discussed, during pregnancy is one of the vital factors for normal fetal development. Early life undernutrition led to various adult metabolic and psychiatric consequences, including atherogenic plasma, lipid profile, cardiovascular diseases, obesity, as well as depression and schizophrenia. Then late maternal age also has an effect. The effect of late parental age on the fetal development is, although not well documented in the literature, there are few reports suggesting epigenetic roles associated with maternal age in the pathophysiology of neurodevelopmental disorders in the offspring. There are several adverse effects seen in aging mothers, such as larger occurrence of preterm deliveries. Late paternal age also is a cause. Altered DNA methylation was reported in aging human sperm, targeting promoted regions of 117 genes, among which few are associated with neuropsychiatric disorders like autism, mental retardation, schizophrenia. And evidence also suggests that sperm epigenetic modifications can survive reprogramming following fertilization and be inherited by the embryo, as told previously for Angelman syndrome as well. Infections. It is possible that maternal infections alter epigenetic mechanism that regulate developmental control genes and signaling pathways vital for normal development of the fetus. Babies with autism associated copy number variations, CNVs, are more likely to develop autism if they are exposed to maternal infection in utero. Urbanization. Research has documented that associations between particular features of urban environment, such as concentrated disadvantage, Residential segregation, social norms contribute to a higher risk of mental illness. Changes in DNA methylation may be one of the mechanisms through which contributes to the psychopathology. Aberrant DNA methylation signatures characterize mental disorders in a community setting. Now, this is also seen in a shift worker. A recent study on a population of night shift workers has shown alterations in blood DNA methylation included changes in ALU repetitive elements methylation and gene-specific methylation of inflammatory genes as interferon gamma and tumor necrosis factor alpha. Aging, it is described that there is a general loss of histones together with a massive alteration in the histone modification patterns due to aging. Now, let's talk about epigenetics and psychiatry. The onset and development of disorders like major depressive disorder, drug addiction, and schizophrenia goes beyond the one gene, one disease approach. Epigenetic modifications, such as, again, histone acetylation, deacetylation, as well as DNA methylation, can induce lasting and stable changes in gene expression. Major depressive disorder, in humans, the genetic polymorphisms in serotonin transporter, gene promoter, can lead to differential susceptibility to the effects of stress, such that carriers of the short allele show a heightened stress responsivity and increased risk of depression when exposed to increasing life stress, childhood maltreatment, or in case of rhesus maternal deprivation. Schizophrenia. Various studies suggest a contribution of genetics to the development of this. However, no single gene has been isolated to its etiology. A hypothesis that emerged regarding schizophrenia was that epigenetic control of gene expression, specifically DNA methylation at promoters within the gamma aminobutyric acid containing neurons, is a key factor in the development of this illness. Substance abuse disorders. Although the path to addiction begins with an individual's voluntary use of drugs, however, repeated exposure can induce lasting changes in brain reward areas, causing individuals to compulsively use and seek drugs. Chronic exposure to drugs of abuse can alter gene expression in key brain areas, including the ventral tegmental area and the prefrontal cortex. Acute or chronic cocaine administration has been shown to increase the overall histone H3 and H4 acetylation, suggesting that these stimulants can influence the epigenetic processes. Suicide. To search for epigenetic markers of suicide, DNA methylation patterns from the prefrontal cortex of depressed individuals who committed suicide were compared to depressed individuals who died by other causes, and only one gene coding for the spindle and kinetochore associated complex subunit was found to be differentially methylated in neurons of suicide completers compared to the control group. Now, in obsessive compulsive disorder, the epigenetic mechanisms play a role in the pathophysiology of OCD. It was reported in females with OCD, differential methylation for GABBR1 gene in blood samples at birth, as well as for MOG and BDNF in blood samples at the time of diagnosis. In post-traumatic stress disorder, evidence indicates that its development is also affected by genetic factors, and research found that genetic and epigenetic factors account for up to 70% of the individual differences in PTSD development, with PTSD heritability estimated at about 30%. Studies have found that stress exposure may even affect gene expression in offspring by epigenetic mechanisms, thus causing lasting risks. Now, eating disorders. An individual regulates weight around a set point could at least in part be due to early epigenetic encoding. Now, 5 genes, namely fat mass and obesity associated FTO, leptin and its receptor, pro-opiomelanocortin, POMC, and BDNF are used as examples that might be epigenetically modified in ways that alter the risk for these eating disorders. Now, if individual with 2 copies of a common polymorphism in the FTO gene has a 70% increased risk of obesity, and a variant and variants of FTO are associated with obesity and increased BMI. In people, early epigenetic changes mediated by FTO-associated DNA methylation might set levels of resting energy expenditure, which could impart a lifetime propensity to high or low weight. If there are societal pressures to be slim, people with FTO variants associated with increased BMI may adopt compensatory behaviors like increased risk of having an eating disorder. Now, in autism spectrum disorder, genomic studies have identified genes that seem to contribute to this condition, and two genes have been known to be associated with it, neurexin and SHANK3. In neurodegenerative disorders, epigenetic mechanisms are essential for normal brain functioning, that is, learning and memory processes, and disruptions of these mechanisms are closely related to molecular alterations associated with disorders such as depression or Alzheimer's. Alterations of levels of 5-MC and 5-HMC and other epigenetic markers during the lifespan have been associated with progression of Alzheimer's. In conclusion, mental disorders have known environmental causes. Also, there is heterogeneity in response to each of the causal factors. The study of gene-environment interaction, starting with early animal studies, documenting gene-environment interaction to demonstrations of similar effects in family, adoption, and twin studies. The initial excitement surrounding this interaction has begun to wear off. A number of challenges involved in the study of gene-environment interaction are now being recognized. These include difficulties with interpreting interaction effects or the lack thereof due to issues surrounding the measurement, scaling of the environment, and statistical concerns surrounding modeling gene-environment interactions and nature of their effects. These are my references. This brings me to the end of my seminar.
You can ask if any of the online participants has any questions.
Are there any questions regarding the presentation?
From here, does anybody of you have any queries you want to put? No? If you don't have any questions.
Ma'am, because folate and vitamin B12, they are responsible for promoting DNA methylation. Now the mothers who are going to be deficient in that, they will not be able to promote the, they will not be able to provide the methyl group that is, that might help in this methylation. So those are the DMD mothers. They are deficient methyl donors. These children will be they have an increased, they might have an increased risk for development.
Can you, in short, just describe concept of epigenetics? What did you understand?
The factors which indirectly modify the genes and which may be inherited and the cause probably can be a reason neurodevelopmental. Those are the factors which may include our eating habits, our stress, our environment, the drugs. But these factors somehow modify the genes in the very basic transcription levels, which further
First slide is shown. Study surrounding stable alterations of the DNA that alter gene expression. Gene is one thing that you have DNA, and gene expression is another thing. So we think that if somebody has a gene, DNA, then automatically you will have that trait. It is not necessary. That gene may or may not be expressed. Okay. So that expression of gene, genotype and phenotype. You may have the same genotype but different phenotype. So that is why even in identical twins, the concordance of the even genetically dependent illnesses, they will have 100% correlation. This is a difference. And this is where environmental factors. Environmental factors come. So even in siblings, there is a difference. Environmental factors it can be apart from factors which Dr. Ishani has said, there is a factor of chance also. Whether it is success or not. That is one. Do you know any differences, like any phenotypes that you can associate with certain with genotypes? There are specific phenotypes, symptoms,
Yes, sir.
some findings where you can say that this genetic represents psychiatry. Is it there?
Sir, in schizophrenia, the soft neurological signs are like jointed eyebrows and high arch palate.
Okay, cognitive symptoms are there. Symptoms that are present in siblings. So, but the expression is not there. So that is also there. Right? It is not. We can find these genetic conditions even in clinical symptoms. Certain things are expressed. Cognitive also some features are there. You can say that this is associated with some genetic marker. But then the whole illness may or may not be present. Okay. So, any more questions? No? Any faculty has a comment? I think
yes, sir.
You can ask if she has any comment about the
Messaging.
It's okay. You can just talk. I may not be audible to her now. I'll talk from there.
Sanjeevani ma'am, if there is any question related to the slide.
Comments regarding the presentation.
Any comments related to the presentation?
The presentation was good, I think. And you have put the references. You have put the numbers at the end. The information could have been a little bit less prepared. Okay? Like if you would have come to me with the presentation, I would have said that maybe cut down on the details. Maybe like when you put that in the slide, it attracts attention, like all the terminologies that you put. Try to limit the number of words that you put in the slide. Okay, you put an image like that, so your slide appears covered. It's an improvement, just a section of improvement. And instead of writing whole sentences, I know that initially you can do that so that you don't stop the presentation, or you can present it quickly. But for the presentation, what you can do, you can have a write-up with you.
Yes.
So what you are going to say about the slides, have it in a card, and on the slide you can put less information. Okay, that is my suggestion. Just for the way. Like we can. Yes.